Okay, now it's time to load the data. As you know, the data is generated by 10x genomics technology, and what we do is that we download the H5 files, and that's how you do it. First of all, I import OS again. I usually try to put all the packages in the beginning of the Jupyter notebook, then we can see all the packages used in the Jupyter notebook or in the analysis, and then we can sort of plan for which type of packages or dependencies we have to do the data analysis. But here I have also used it here, but that's just a minor detail. And then we have the path to the data. This is like where you are going to download your data, and that's just a URL. And then here we specify at which path we would like to save it. You can name it as whatever you want. It is just a variable name that we have chosen. So what we do is that we ask whether this directory exists or not. If not, then create this directory. Then we also create another folder for the results. We do the same procedure and create a new folder which is called data COVID results. And then we import the URL lib in order to download the files or the data from this URL. And the files that we are gonna work with are these that are in this list. 1, 2, 3, 4 normal samples, and 1, 2, 3, 4 case samples. The way that we do it is that now we go through each of the elements of this file list, and then we specify where we will have these files. So just for sanity check, if you want to see what is really happening under the hood, we do like this. So if you want to use the variable names while you're printing a message, you can use the F just before the quotation, and here I say path file. Sometimes this auto completion by Copilot is very aggressive. Maybe I can pause it for a while. So here I come, I press this one and say okay, snooze it, for example, for 1 hour. Okay, there we go. So what I did here was to print a message in order to see at which path my files are going to be written. And in the next if condition, what I am trying to do is that to check whether this path exists or not. And if it did not, then it creates a new URL, generates a new URL. So let's do like this, print f, and then I say I am downloading file URL. There we go. So then we will see what is happening under the hood. So just for sanity check, this is the folder structure that I have. As you can see, there is no other folder, and we will see upon running this chunk whether we will get these folders or subfolders as well or not. So there we go. So yes, we have the data and then COVID and then results. And here you see that we get the path to the file of interest or dataset, and then here it also shows us which file is being downloaded. This file will be downloaded and then saved as this file name, and so on. Now that we have downloaded the files, we should upload them in the Jupyter notebook that we are working now. So the way that we do it is to call the method or function called read 10x h5 from sc. So sc is the Scanpy that we imported first. So we said import Scanpy as sc. That's how we get access to the methods or functions. And then we use OS path join. It's just to sort of put together the file of interest under given path and then download it directly. So instead of specifying the path manually, we use this because it will help you when we go downstream of analysis. Instead of copy pasting the path, you will have this structure, and this can be also delivered to your colleague or in your team, so it will be a bit more reproducible instead of having path, for example, desktop Nima but here you are working within the folder, working folder, and then all of these files based on relative path will be accessible. So it was probably a bit too much detail, but what we are trying to do here is to load the data, and then we make the variable, the names to be unique. So then we will have one variable or one object for each of the samples. It gives you one reading which file and so on. And then we also add some additional information. So before going into that, let's look what each of these files look like. So let's say data underscore cov1.obs, not obs. So what you see here is that these are the structure of the data that we have within the sample. The number of observation are it's 1,500 cells, rows, and the columns are in var, which is 33,000, corresponding to the number of genes that we have. Okay, in addition to this size that I just told you, we have the var slot. So if I check var and then just hit the button, Control Enter, or pressing this play button, what I see is a data frame like information which consists of gene IDs, feature types, and genome. And then on the row you also have the gene names. So if you go to the AnnData object structure, what we see here is this one. So under observe we have the rows which corresponds to the cells, and then on var we have the gene information. So we can also check what we have in the observation slot. So if I press ops, then these are the cell identifications, which we have 1500 rows. Now let's add some additional information. So for example, we would like to give each of these cells their type and also their sample information. So if I hit a button, we can check what we have in the ops now again. So there we go. You see, for each of the rows now we have added the type and also the sample. So if you want to see whether the information has been properly inserted, so I would say the type. So there we go, we have the COVID, and now I would like to check the sample ID. There we go, yes, so it's a match. You do not need to do that, it is just to showcase you what we have done to the observation slot. Anyway, now let's merge all of these data sets together, and the way that we do it is by a concatenate method. So in AnnData, when you have AnnData, you already have access to some of the methods. So what we do here is that we say, okay, we would like to concatenate these files or these objects and data objects to data cov1, and then we store it as a new variable. So at the end we will have one single AnnData, and then we delete the rest of them, which includes of the cases and also control. So then there we go. So let's see what we have in our AnnData now. So you see now we have 12,000 cells with 33,000 columns or genes, and then we have two slots, we have observation, and also we have var. So let's look at what we have in the observation. So you see now we have all of them together, all the cells together with their types and also the sample and which batches they have been labeled. We already answered this question which is asking us how many cells and genes are in this data set. Now let's check out I cannot talk. Anyway, we can check the var as well. So what we have, again, we have the gene ID, feature, and genome, which we looked into that.